Welcome to the third lecture on molecular genetics. This section is going to cover mutations. I'm Wendy Nofke and I will be doing your presentation today. Mutations are a change in the sequence of bases in the DNA or RNA. And mutagens are the substances or environmental factors that are going to cause these mutations or changes in the RNA or DNA. These can be physical examples, chemical agents, or infectious agents. Some examples of physical things that can cause mutations would include UV light and X-rays. Chemical agents include things like nitrates or nitrites that are used as preservatives in food, benzoyl peroxide, some of the chemicals in cigarettes, and one of the more common things is the smoke in barbecued foods. When the fats drip down onto the fire portion of the barbecue, it will actually form carcinogens in the smoke that will actually come up and land on your food. And then there are infectious agents like the human papillomavirus that will also trigger mutations in your genetic material. The effects of mutations can depend on where they come in and occur and how early they occur in the organism. Germline mutations are going to be ones that occur in the gametes or your sex cells. These can be transmitted to the offspring and all of their cells. So if an organism is fertilized with a sperm that has a mutation in it, all of their cells will carry that mutation. The somatic mutations tend to have a little bit better outcome. They're not in the gametes, and these don't get passed to the offspring. Instead, they're confined to the one cell and all of its future daughter cells. We can have alterations of the chromosome occur on a larger scale. These will change the chromosome structure, and these tend to be very serious. In a lot of cases, they're actually fatal to the organism. When a deletion occurs, a portion of the chromosome is removed. With a duplication, part of the chromosome is copied, and then it's inserted on the other homologous chromosome. With the inversion, you have sections of the chromosome come out and insert back in, going the other direction. With an insertion, part of a chromosome is removed, and it adds on to the homologous chromosome that it's paired with. With the translocation, part of the chromosome is removed, and it adds on to a non-homologous chromosome. This diagram demonstrates some of the differences. Here, where you have the deletion, you have what started out as the normal size chromosome and a smaller one as a result. With the duplication, you have this region of it that becomes copied and duplicated and is now twice located in the chromosome. With the inversion, you can see you start out with the region going in one direction. It comes out, turns around, and goes in the opposite direction. With the insertion, you have part of the chromosome come out and it inserts onto another chromosome. And then with the translocation, you have this region that comes out and it will attach to a different non-homologous chromosome. We can also have point mutations, which are changes in just a single nucleotide in the DNA. These can have varying effects as well. The silent mutation is going to code for the same amino acid. There will be no change in the phenotype or the appearance of the organism. This is often the best result because it will code for the same exact thing. The reason this happens is because there is the redundancy in the genetic code. And here is an example of a silent mutation. You can have a change here and it's still going to code for the same amino acid as it did here. With the missense mutation, it's going to end up coding for a different amino acid. This can have a variable effect. It could make a protein that's better, one that's worse, or it could stop it from being made at all. The nonsense mutation is going to code for a stop codon. This ends up terminating the synthesis of the protein early. With the Frameshift mutation, you have a deletion or insertion of one or more nucleotides. This is going to change the reading frame of every codon downstream from where that insertion or deletion occurs. These can have very drastic effects because every single amino acid afterwards could be altered. The effects of mutations can vary. They can be neutral, where they have neither a positive nor a negative effect on the organism. Many mutations are actually repaired before protein synthesis occurs. You can occasionally have beneficial mutations that will lead to a new version of a protein that actually helps the organism. An example would be bacteria being able to survive in the presence of antibiotics. It's not beneficial to the host that gets them, but it is beneficial to the bacteria that they now have a new gene that is able to survive in the presence of that antibiotic. Some are actually harmful to the organism as well. Most mutations actually do end up being harmful. There is a very small chance that a random mutation would actually benefit the organism. It's far more likely to cause damage. Some examples would be genetic disorders, where you have a disease that's caused by a mutation in one or more of a few genes, such as cystic fibrosis. Another example is cancer, where cell growth becomes out of control and forms abnormal masses of cells. Most cancers are usually caused by mutations.